fast and war is a combo that has accompanied human history for a long time, so much so that I often say that horses have been the animals that have been the most influential in human history because they have been with us for as long as we can remember until the start of the 20th century. But questions as simple as when did we domesticate the horse from the land? Where did that happen? How was domestication? And how did we even succeed into transforming a wild animal into this so important animal for us were questions that were actually unsolved until our work. And what I'm going to cover today is how did we manage to use ancient DNA and ancient genomics, in fact, that is sequencing the DNA and the genome of ancient horses as discovered in the archaeological record, combining with the genomic evidence from horses living today to reconstruct the whole area of horse domestication. So our first test or experiment that we conducted was conducted in Kazakhstan, in the north of Kazakhstan, in a place called Butai. And this place is what you see here. That's an excavation that is named Butai. That dates back to 5.5 thousand years ago. You see that this is the step from Kazakhstan in Central Asia, and what you see here, this round structure, is a house of people that were living at that time. Okay? And they had circular houses, and you see that archaeologists they discover some bone accumulated in pits around the houses, and in fact, what you don't see here is that there's a whole village of Butai people in this region. The reason why it's important to be interested in that region first is that if you're an archaeologist, you will realize that this is where the first evidence for horse harvesting, for horse milking, and for horse foaling, which is putting the horses in enclosure, that has been discovered ever in the archaeological record. Since it's the first time in the archaeological record that you have evidence of us domesticating the horse, putting the horses in enclosures next to our houses in big numbers, our hypothesis was that it may be at Butai that everything started. And so our hypothesis was if we were to sequence the genomes of the Butai horses, then we would have the ancestor of all the domestic horses that have ever lived on the planet since. We would have the starting point of everything. So if we had the genome of Butai horses, then we could understand the very first genetic modifications that humans would have selected to transform the horse from a wild animal into a such important domestic animal. And so the only population of horses that would have been not related to horses that still exist on the planet would have been what we call the Przewalski's horse, which is native of Mongolia and Inner Mongolia. That is the single horse that considered still alive to as the last full wild horse living on Butai. And we sequenced 25 genomes for Butai horses. There's hundreds of different genomes post Butai, after Butai, across Eurasia, spanning the last, in orange, 4.2 thousand years, whereas Butai is 5.5 thousand years ago, as I told you before. And the result we had was nothing like we expected. Because as you can see in this population graph that is the best fit to our data, is that the Butai horses, those that are supposedly the ancestors of all the domestic horses living on the planet today, are actually directly ancestral, not to the modern domestic horses seen in dark orange here, but of the Przewalski horses. So what I tell you here is that, yes, we domesticated the horses first at Butai, but that horse did not give rise to modern domestic horse diversity. It gave rise to the Przewalski's horse, which is the horse that I is the last fully wild horse living on the planet. So what it implies is that the Przewalski's horse is not white. He's actually the feral descent of the first horses that have been domesticated. And this fertilization process probably started over 5,000 years ago, which is the age of the last Butai horse that we have sequenced in our data. But the second implication is more important in my opinion, is that it also tells you that the modern domestic horse that we know of today actually started somewhere else, in another place than Butai, which remained to be discovered. So what that word implied is that there were two domestication processes for horses. One at the time, this one resulted in the Przewalski's horse, Bufferization, and another one somewhere else in Dom 2 was the second domestication process that gave rise to the modern genetic diversity, and also to the diversity of domestic horses that we had in orange in my previous slides, which are the horses that have been domesticated and have been recognized as such for the last 4.2 thousand years in history. So when we had these results, we were very excited, and we spent the last 5 to 6 years to actually do some more to try to identify the context in which the southern domestication process took place. Oh, and by the way, genetically, two things are independent because we could try to estimate the amount of gene exchange between the two domestication processes, and we found a median estimate at 2.7% only, meaning that DOM2 are 97.3% independent genetically than the type. It means historical independence, something totally different. So we did a lot more sequencing across Eurasia and sequenced not just bow tie, but everything we could get access to try to see where bow tie, where DOM2 actually unfolded. And what we found is answers to all the questions that I mentioned. So here I'm reconstructed based on the genetic variation, the variation of the size of the population, and E that we saw before in my previous lectures as a function of time. And here time 0 is 1,000 years, 800 years before the Common Era. So that would be 3.8 thousand years ago, if you will. So we look at the variation of the size of the population from 3.8 thousand years ago and before. And what we saw was very surprising is that there is a time period when there is almost no horses available to us. This particular moment where there is only a small stock of horse, we interpret it as a domestication bottleneck, the moment where people started extinction that is really, really fast, almost taking place in 10 generations, which for a horse means 80 years, that actually expanded the population of horses everywhere. So we know that as far as the when is concerned, when horses have been domesticated is during this bottleneck period, because after that period, they are expanding in big numbers. So we put a time on the domestication of DOM2 anywhere between 4.2 thousand years ago to 4.5 thousand years ago. So as far as the when is concerned, we have an answer. 4.2 thousand years ago to 4.5 thousand years ago. But how about the when? The where, sorry. And the where is because I'm showing you here the population structure that we unfolded before DOM2, before 4.2 thousand years ago, where each chart that you see is a pie chart providing you the genetic composition of the underlying population. Colors represent different genomic ancestries, if you will, and the size of the pie chart is the number of horses we have sequenced in the past, right? So what you see here is in different regions you have different colors. For example, in Botay you have green dominating, whereas in France you have light blue dominating, whereas in Siberia you have purple dominating. So what it tells you is that different regions have different lineages of horses in that. And because that's the case, it should give you an intuition about what it means, which is that if there is a diversity of colors in different regions, it means that those regions are isolated from each other. Otherwise, they will share the same genomic ancestry. So as genetic population geneticists, you could actually use this map to predict the map of gene flow across Eurasia at the time. And so this is what those ribbons show you, and when the ribbons are very brown, it means that the populations are isolated. They don't migrate. They stay where they are. So that's the situation before DOM2. But what happens after DOM2 is that it's something totally striking. So I invite you to look at the screen now, and this is the situation before DOM2, and this is the situation after DOM2. You see only one color, ancestry, expanding across Eurasia overnight, 4.2 thousand years ago. The orange color is everywhere. So indeed, the when is really 4.2 because this is the key moment where everything changes, basically. But now we're left with the where question, which is an interesting question. So how did we solve that? Well, we have the map of the population structure before. So what we could do is train neural networks through the locator spatial correlation software to learn what is driving population structure genetically. So in other terms, you train the neural networks to learn which genetic mutations are actually responsible for this genetic structure that you see. And then once you have trained those models, you can take any of the DOM2 genomes that you have since, from 4.2 thousand years ago to today, and ask a very simple question. Is where those ancestors were living in the previous map? And this is the pseudo-livelihood surface that we reconstruct when we do that. The several hundreds of horses we have sequenced in the last 4.2 thousand years actually already know their ancestors come from this particular region on the map, which is the north of the Caucasian Range, north of the Caspian Sea, somewhere in western Russia. So that solved the where questions. We have revealed genetically the homeland of the DOM2 domestication, which is independent both geographically, temporally, and genetically to the multi-domestication process. So since we have solved the where, we've been interested in a number of questions such as how long did the local lineages survive until today? So to exemplify this question for you, I'm actually inviting you to have a look here. As I told you just before that the DOM2 expands everywhere, but you realize that when they expand, there are places where other colors remain. For example, in Spain, where you have this blue thing or purple thing that used to be there, that's still surprisingly present as they expand everywhere else. So we have a follow-up study now where we are looking at what is the process going on in Iberia, for example. So we sequenced in Spain and Portugal tons of genomes of horses from 24,000 years ago to the 26,000 years ago, to the modern times today. And we track the chances of the gene composition through time in Iberia specifically. And long story short, we discovered that in Spain and Portugal, there was a yet different lineage of horses that used to be there. We call it IB for Iberian, Ibe, here in pink. These are the DOM2, very different genetically. These are botai, so those three things are very different from each other. And what we learned is that this Iberian lineage not only existed in Iberia during the last glacial maximum, so before the Ice Age, if you will, but survived in continuity in Spain and Portugal until pretty much 571 years before the Common Era, until 2,500 years ago. Our studies have also revealed that the first DOM2 entering Iberia in our lab happened to enter in Iberia about 3.1 thousand years ago, 1.1 thousand years before the Common Era. And we did some more work because some of the horses we found there are actually a mixture between the DOM2 and the Iberian. So between the DOM2, pure DOM2, no, a pure Iberian, but it's more so the mixture of 80% DOM2 ancestry and 20% Iberian ancestry. So when you have such horses, what you produce is softwares to date the time of their mixture. And when we do that, so I'm sorry, I should have said that all those horses, they are confined into Iberia and southern France. So really the Iberian lineage was indeed local, but when you use software to estimate the date of a mixture, you find three things. The first is that the most recent date of a mixture was about 2.3 thousand years ago, 300 years BCE, before the Common Era, which means that the local lineage in our data set became extinct 2.3 thousand years ago. So it takes 1.7 thousand years ago for that lineage to disappear, basically. It becomes extinct indeed. The second thing is that you see that there are many days of admixture. So it means that the admixture was continuous with the DOM2. You know, it means something very important. It means that the breeders of horses in this region were permissive to hybridization with the wide local lineage. They don't isolate their horse from the wild horses. Second information. The third is the most important in my opinion, is that the earliest dated estimate for the admixture time between Dom2 and Ibe, the local lineage, dates back to almost 4,000 years ago, 1.995 before the Common Era. So that's almost 4,000 years ago. Which means, in fact, that if horses were domesticated in the steppe 4.2 thousand years ago, it took only 200 years or 250 years for them to actually reach Iberia. But our work has also, so it means nothing. It means in 2 or 3 centuries, they travel all across Europe. Our work has also revealed in a paper we just released 2 weeks ago in Nature that it took only 50 years for horses to actually come across the Carpathian range in this heart of Europe. And we also showed previously before that it took only 100 years for horses to actually reach the Altai. So what I'm telling you is that when the Dom2 actually were created as a breed, as a bloodline, they actually spread across Eurasia in no more than a few centuries and replaced the local lineages very rapidly. So the question of the how is the last of the questions that I have to leave today. How come we managed to domesticate with success Dom2? And I invite you to think again about this expansion process and bottleneck that I've mentioned before. So the how question we have actually divided into 2 sub-questions. The first is how come during this bottleneck people have, which are the genes that they have selected, transform this new bloodline? Is there a certain number of domestication genes that we could uncover? First question. And the second question is how come can we even from so little number of horses multiply them in so high numbers so that they invade and replace all the horses at the scale of a full continent? This is not obvious. So we have, I'm lucky to tell you that we have solved those 2 questions now. The second question we've solved by inventing a new statistic to measure generation times with ancient DNA data. So the principles are very simple. Imagine that you have a tree. You remember my lecture about tree thinking. Trees are very important. So imagine that you have an outgroup and a series of horses for which you have radiocarbon dates. You know when those horses were living. If everything is the same population, you will have a tree like that. They are spread across the tree as a function of time. If the mutation rate is constant, then you will see generation time is constant. You will see that the horses that will diverge first will be the oldest, whereas the ones diverging last will be the most recent, simply because they accumulate differentiation through time. So if the generation is constant, if you calculate the distance from any of your horse genome to the output, the distance will simply increase as the radiocarbon date estimates goes more recent. Okay? Very easy. But if the generation time changes, then mutations will accumulate per unit of time faster. Because what creates the mutation is each generation, is each meiosis at the time when you create spermatozoids and ovocytes. So if you see an acceleration of the distance to time, then it tells you that during that period, generations of horses tick faster. The generation clock was faster. And this is the stuff that we have tested, and this is what it shows you here. In other words, that the breeders put to reproduction their horses much earlier than they would have in the wild condition. And you see that what we found in our latest analysis is that there were 2 moments, in fact 3, but 2 moments where in the past horse generation times were reduced. This is 4.2 thousand years ago, which is the, and this is 5.5 thousand years ago, which is the Botan event. So typically when people domesticated the horse twice, they did so by controlling the reproduction of the animal and by becoming able to put mares and stallions to reproduction earlier than they would have in their life, so that their generation time was actually absolutely accelerated. Why did they do that? Simply to produce more horses per unit of time. different generations. If you have 2 generations in 10 years instead of 1, you will have every 10 years 2 to the power of more horses available as a breeder. So very fast you can produce big numbers to invade horses the whole continent. Interestingly enough, we found an acceleration of the generation time also 200 years ago in the 19th century, which is documented as a time when breeders have nibbles with the reproduction of the animals as well to create the modern breeds that we know of today. So we're very confident now. The second question about the how is really, can we identify a gene that we have selected early on that explain why DOM2 and no other horse has been so successful? And the short answer is yes. We answered that by doing a very simple test that we actually learned how to do over the last few days. We calculate FST, you remember this fixation index that tells you how different 2 things are genetically. So we simply calculate FST between 2 groups, the DOM2, the successful ones, and all the other lineages that have ended up being replaced by the DOM2. And in that graph, you see the 31 autosomes of the horse, and every single point is a SNP. And when the y-axis goes up, it tells you that the 2 groups, the DOM2 and the non-DOM2s, are very different genetically. It strikes you potentially that on chromosome 3 on the one hand, and on chromosome 9 on the other hand, you have 2 places in the genome where gene variation is segregated very differently in the DOM2 and the non-DOM2. So for us, it was a lead that potentially there are genes that have been selected in the DOM2s so as to allow them to become domesticated. So what are the genes lying there? So one on chromosome 3 is LFBN1. That's a gene that is very well known from experiments, behavioral experiments in rodents, especially in mice in that very case, where you put mice on a sort of cross, where 2 parts of the cross are protected by walls and 2 are in the wide open. And you simply quantify using CCD camera the time that mice spend in the open versus in the protected area. And this is a very typical test for assessing the behavior of the mice, especially whether mice are risk adverse or risk taking, or whether they are aggressive or docile actually. And so you realize that when you KO, when you knock out ZFPM1, that very gene, you see the behavior of the mice changing both in males and females, by which the inactivation of the gene really makes mice spend a lot less time in the open. These tests are typically interpreted as a change in behavior of the mice. So clearly we know that ZFPM1 is a key regulator of mice behavior, and it's a key regulator of mice being able to take risks, and mice being docile or aggressive. It can be demonstrated in many, many different ways. So we interpret our results as ZFPM1 being one gene that has been selected early on because potentially the variant that was selected created a new type of behavior for horses, a behavior that was probably more docile, less aggressive, which makes sense in the context of humans interacting with the animals, and the animals being parked in enclosures. They will have to be less aggressive to each other and to the humans that interact with them. So behavior was controlled through a selection of ZFPM1, and that is probably one of the key features that allow horses to even be domesticated. The second locus that we have identified was chromosome 9. At this time on chromosome there is a gene, GSDMC, who's, and I can't have the time today for demonstrating that, but which is responsible for a very important change in locomotion in the animal. We know that now from lots of experiments, but we discovered that gene based on a pin experiment, which was, if you look at GSDMC in humans, you see that the variation of the gene is associated with what we call the spinal lumbar stenosis, which is a defect on the spine, basically, for humans. And humans who have a particular variant of the gene will have pain as they walk, so much so that they can't walk anymore. And humans which have a different version of the gene, they are like you and myself, they don't care, they can walk. Right? So we interpret that finding as a key modification of the gene has potentially changed the horseback anatomy, allowing horses to be more mobile than they were in the wild, successfully making them useful animals for us. So what happened after 4.2 thousand years ago? A lot, and I don't have the time to cover everything that we have done, but I'm going to take one example to tell you how ancient genomics has been really revolutionizing our ways of thinking about the past, not just the past that's deep 4.2 thousand years ago, but even the most recent past. And the example of the most recent past that I'm taking now with you is the colonization of the Americas. Because humans colonized the Americas in 1492, and the picture of the Americas has been changing since. So the question that I'm going to focus on now is the horse that is the horse of the indigenous peoples living in the Americas. What is the origin of that horse? The reason why I'm interested in that question is that there are many ways to think about the question, but the most common model for this, for the origin of the horse in the Americas, is to believe that the horse became extinct about 20,000 years ago in the Americas. We know that from paleontology. I've told you just before that with environmental DNA it might be down to 8,000 years ago, but eventually becoming extinct in the Americas. So because it became extinct, your horse in the Americas can only have sources in Eurasia. And these horses from Eurasia would have been brought by the conquistadors in 1510, and then would have started expanding in the US, well, in the Americas, ending up being passed to indigenous people from 1680, which is the time when there has been a massive revolt of the Pueblos in New Spain and New Mexico. At the time, the indigenous people took over the power for 20 years over the conquistadors, took their hands on the horses and distributed the horses across the place. So that's the model that we wanted to test. Long story, sorry, I don't need all this. Long story short, what we've done is simply sequencing many horses living today with indigenous people in the US in pink, but also sequencing in red horse genomes, historical horse genomes from the Americas dating back to the 16th to 19th century. And what we found is very striking. First, if you use radiocarbon dating of the horses that we find there, we see that in different regions, we can date that horses existed in the Americas already by 1600. No big news because the conquistadors came with horses, we know that, from 1510. But what was more fun is that if you look at those horses, some of those horses that are very early 1600 actually were fed with maize. And if they were fed with maize, it means those horses were indigenous, were with indigenous. Because maize is an indigenous plant. No Europeans ever grown maize before being brought by another horse, because we see that with this fracture that is part of the soil. And we will survive the fracture because it was resolved at the ossification level, was fed by indigenous people and cured by indigenous people. It means that year 1600, horses were already with indigenous people. So that means that we had to revise the model that I told you about before. It didn't take as long as 1680, because by 1600 already, indigenous people had access to horses. But if you look genetically at those horses, mitochondrial DNA, Y chromosome, or even the whole genome, what we found is that those horses that were in red, part of the indigenous people, and those that are in pink, part of the indigenous people today, as I'm speaking, they all belong to the Bantu lineage. They are all Bantu. None of them shows any significant amount of genomic ancestry coming from the horses that lived in America before 12,000 years ago, which is shown in orange. Which means that those horses indeed are European. They are not the descent of the horses that used to be native from America more than 12,000 years ago. We could actually demonstrate even something more striking to me is that if you calculated how close the horses that we sequence historically or today in America are to Spanish horses, or how close they are from British horses, you realize that the historical horses we had were more Spanish than British, because they are attracted to the Spanish component. Whereas that was from the 16th century to the 19th century. But if you look at the horses living today in the Americas associated with the indigenous people, they are more British. So what you show here is that the colonization process has brought first horse bloodlines from Spain and Portugal with the conquistadors, but as the power shifted in America to Britain, because Britain creating the USA, right? They brought new horse bloodlines from Britain, from Northern Europe, shifting the whole composition, or genetic composition of horse populations in the America. So what we measure is that the colonization impact did not just restrict, but actually extended to the genomic history of horse. Colonization is indeed a very powerful force. And this is the model that we published, in fact. So yes, horses seem to have become extinct in the Americas, even though they are native from Americas. It's not the Viking who brought the horses in America and conquistadors, but so there is no news there. But the big news is that the horses went to the ends of the indigenous peoples as early as the 16th century. My conclusions are very simple. This is the diversity shown as a principal component analysis here. This is the genetic diversity of horses that used to exist before 4.2 thousand years ago. Each point is a genome that we have characterized in our work. You see that the diversity is very large. What I invite you to look at now is the screen about what is left today. So the domesticated horse has completely reduced, almost annihilated the diversity that used to exist even 4.2 thousand years ago. The diversity that exists today is really nothing compared to the diversity that used to exist. So yes, it shows the impact of our activities on biodiversity, because we lost our existing diversity simply by domesticating them. That's the first element of my conclusion. The second is that the horse is not just a biological thing, it's also a social thing. Because if you think about the horse, these are also the animals of the kings, of the queens, of the elite people in society. So there is a social dimension we never forget about when we think about horse evolution. And I'm going to spend 2 minutes to show you how we could still use DNA to document some of the social and consequences of our relationship with horses. And I'm going to do that with the nomads living in the Iron Age in Central Asia, the Scythian Pacific nomads, not far from here actually, because those people were really horse masters. They have left very, very world famous goals representing their horse skills. And the question that we were interested in is what kind of social impact that the horse had in their society. So we went to develop a society in Kazakhstan in the foothills of the Altai. This is how the site looks like. You see an accumulation of stones. If you dig below the stones, 6 meters below, you find the coffin of 2 people. But more interestingly for us, here specifically, you see skulls of horses. In fact, you see full bodies of horses next to the coffin, and those horses come in 2 layers, 6 plus horses on top of each other. And when you look at the skulls, they are well preserved, but you can also, they're very well preserved. You see some are dressed with gold, also the jaw, of course. But you could also see that on the skull, they were killed on spot, because you see a hole in the skull showing that they were sacrificed. So the sacrifice is a very strange practice, but it's a very common practice in many parts of the world where animals were killed at the time when very important person dies to help the animal bring the person to the afterlife world space. So there is a very strong social component about what animals you kill and to actually help the dead person to actually transit into a new world, right? So we ask the question, can we with DNA identify which kind of animals they killed? And so what we learned is that all the animals were males. They never sacrificed females.
Oh.
All the males were not castrated. None of them was castrated. They were full. They were killing horses of different age, from 2 years old to 21 years old. So that means that they did not just sacrifice the old, those that have no economical values. They were actually killing across the whole age class. And we showed that except two, none of them were related. So it tells you that they were not breeding a special family of horses for sacrifice. And so you see how DNA can tell you a range of information that archaeology would not have unfolded without DNA, that could be very informative. The social norms, by that I mean the social constraints, the social habits that people had in the past in their relationship to the animals. And since we were successful in doing that at the helm for 13 courses, we'll be able to show, by the way, that they were all different kind of colors. They didn't kill a special family of a particular color. They killed across the diversity of colors. So since we could demonstrate that for the helm, now we have embarked on a very ambitious project, we call it Horsepower, which we will run until 2029, in which we are doing the same type of approach on Mongolian sites and Chinese sites, in fact, where you have not 13 horses sacrificed, but hundreds, if not thousands. So here you see the tomb of one person, but you see satellite tombs that if you excavate them, you see a horse skull in each and every of those satellites. So in this single site, you have more than 800 horses that were sacrificed. And we want to sequence them all to actually unveil that we're actually guiding these practices and better understand the relationship of the humans and their animals at that time in the past, which is for that picture about 3,000 years ago. So thank you so much for your attention today and for your attention over the last 3 days. I really understand that it was a lot, and I'm sorry for concentrating so much